open my slides. Yeah. Okay, so I'm going to talk about identifying coding long non-coding RNAs using deep learning training dynamics. And this work was done together with Berke Dilekolu under the supervision of Ogün Adibili and Öznur Taştan at Sabancı University. I'll dive right into the problem definition. So this gray circle here represents just a small question. Can you see my cursor or not? I am guessing not.
Yeah.
Sorry, just a second. I'll turn the drawing on.
Right now you can draw, I guess.
Yeah. Okay, so this gray circle here represents the complete human genome, and a large portion of the human genome is transcribed, so it's converted into RNAs. So that's represented here as well as here. Only a small portion of the RNAs are translated, and these are the mRNAs. But a large portion of these RNAs are called non-coding RNAs, and these include stuff like miRNAs or tRNAs and all sorts of other RNAs, but also a large class of RNAs known as long non-coding RNAs. So these are defined as non-coding RNAs which are longer than 200 nucleotides, and these are what we are interested in. So the problem is that some long non-coding RNAs, they actually contain small ORFs or open reading frames, which code for small proteins or micropeptides. And I put some examples here of papers showing this. So some micropeptide encoded by a long non-coding RNA regulates muscle performance, suppresses colon cancer growth, define a new eukaryotic gene family, control cell fusion and muscle formation. So why did we miss these before? We missed this before because the traditional criteria of what an ORF is that an ORF has to have 100 amino acids in eukaryotes and 50 amino acids in bacteria. And because of this, this criteria excludes some non-coding RNAs that contain much smaller ORFs. So these ORFs have length less than 100 amino acids. But as I have shown you in the previous slide, there is growing evidence that some non-coding RNAs produce biologically relevant micropeptides, and people have shown this is true across species, so in humans, flies, and in bacteria. So what are the experimental techniques that we can use for discovering these coding link RNAs? So ribosome profiling is one, and mass spectrometry is another. I've put a figure here to show you what ribosome profiling is. So when RNAs are translated, they are bound to ribosomes. And what people do is they get a cell extract and then treat it with nucleases. And what happens is nucleases that are not bound to ribosomes, they are digested by this nuclease treatment. And then if you purify for ribosomes, you can get the protected nucleotides that were bound to the ribosomes, and you can extract these, prepare a library, and sequence them, and you can find out which RNAs were bound to ribosomes. So it's like a proxy for which RNAs were being translated. So the problem with these experimental techniques is that they are cell type dependent. So if you want to know every possible coding link RNA there is in the genome, you would need to sequence a lot of different kinds of cells under very different conditions and so on. It's expensive to perform, also time consuming, and in case of mass spectrometry, it's not very sensitive. So the aim of our work was to develop a computational approach for discovering these coding long non-coding RNAs. The idea is that we already know most of the sequences of these long non-coding RNAs, and if we can figure out which long non-coding RNA is actually coding just by using the sequence, then we don't need to like perform experiments on many different kinds of cells to be able to figure out which long non-coding RNAs are coding. So here is how we do it. So the first step is that we have a data set of like 38,000 coding RNA sequences and 19,000 non-coding RNA sequences. So we perform some pre-processing, and coding RNAs are represented in blue and non-coding RNAs in orange. So what we do is, for every RNA, we represent it as a matrix of numbers. And how do we get from like a nucleotide sequence to a matrix of numbers? This is how we do it. So we have a nucleotide. We have a nucleotide sequence. We get sliding window trimers from this sequence, and then for every trimer, we get an embedding from a pre-trained model. What this means is that for every trimer, like ATG, we have a 100-dimensional vector that represents this trimer. And this way we can get this matrix of numbers to represent the nucleotide. And we also know that, like with every matrix of numbers, we also know the ground truth label. So we know that this represents a coding RNA and so on. What we do then is we take this matrix of numbers, we put it into our deep learning model. We ask the model to predict the probability of every RNA being coding or non-coding. And once we get these predicted probabilities, we compare them to the ground truth labels that we have. And we get the difference between these two. And then we can backpropagate this difference or loss to train our model. And we do this whole process many times. So we do this for multiple epochs. And what kind of deep learning models are we using? So we have 3 different kinds. So the first one only has like 3 different CNN layers. So we call this CNN model. The second one is pretty similar to the first one, but it also has a bidirectional LSTM layer. So we call it the LSTM model. And the final one has a transformer block only. So we call it the transformer model. And each of these models takes as input a nucleotide sequence and then gives you an output probability saying that I think this is 0. I am 80% sure that this is non-coding, and I think with 0.2 probability that this might be coding. Okay, and one final thing that we need to know from this slide is that every time we ask the model to predict probabilities, we also save the probabilities at every epoch. So I will show you how that comes in useful in the next slides. Before we do that, we measure how good our models. So I told you we have 3 different models. So we measure how good they are at predicting whether an RNA is coding or non-coding. And they're pretty good, like AUC above 90 for all of them, and area under precision recall curve also above 90. So these models are pretty good at knowing the difference between a coding and non-coding RNA. And I told you here that we save the predictions for every epoch, and now this is what we actually use for identifying the misannotated lncRNAs, and this figure shows how we do it. So on the y-axis we have the estimated coding probability, and on the x-axis we have different epochs, and every line here represents an RNA. So let's consider RNA A. Also, one other detail, the colors represent the ground truth label. So let's look at RNA A now. We see that across different epochs, it always has a high estimated coding probability. So the models consistently think that A is a coding RNA, which actually makes sense because the ground truth label for A is also coding, so it's in blue. And if we look at E consistently has a low estimated coding probability. which means that the model consistently thinks that E is a non-coding RNA. And this also makes sense because E is in orange and the ground truth label for E is non-coding. So A and E are fine. But what we are interested in are RNAs like B. So B is represented in orange here, which means that the ground truth label for B is non-coding. But if you look at the estimated coding probability, it's always high across different epochs. So the models consistently think that B is a coding RNA when in fact the ground truth label for B is non-coding. So these are the kinds of RNAs we are interested in, we are trying to find. And then I put C and D here just to show you that like the model can change its mind. So like D starts off with a very high coding probability, but as the model is trained, we ultimately end up seeing that D is not actually a coding RNA. All right, and so what happens next is we take the mean. Let me use this, but decrease the brush size. Okay, so I just showed you this figure. Let's consider A again. So if we take the mean for the coding probability of A and the standard deviation, then it always has a high mean and low standard deviation for its coding probability. And if we consider E and we plot its non-coding probability, then it has a low coding probability, which means it will always have a high non-coding probability, and it doesn't change very much, which means that it will have a low standard deviation. So what we do is we plot the mean for the ground truth label and the standard deviation for the ground truth label. So RNAs like A and E will fall somewhere here. They'll have a high mean for the ground truth label probability and the low standard deviation for the ground truth label probability. But if we consider B, and the ground truth for B is non-coding, and B is consistently predicted as being a coding RNA, so it will have a low predicted non-coding probability and also a low standard deviation because it doesn't change very much. And then RNAs like C and D will fall somewhere in this region. So this is just to show you that to identify RNAs like B, we can just take like the RNAs which are in this hard to learn region and then analyze those. What's also cool is that this graphic is again colored according to the ground truth labels, and it seems that most of the RNAs for which our models think that the labels are incorrect are non-coding. So most of the stuff here is in orange. Yeah, and once we get these RNAs in this hard to learn region, we can filter for long non-coding RNAs, and then we analyze those and see what happens. So that's the basic idea. And again, we compare our 3 different models, and now we have this list of lncRNAs that the models think are actually coding. And if we compare it, we see that there is a high overlap between the models, which is good because these are different architectures and they seem to be agreeing to some degree on a bunch of RNAs, on a bunch of our lncRNAs being non-coding, being coding. And then we also make a comparison to CNCRNA database. So this stands for Coding Non-Coding RNA Database. And they describe themselves as a manually curated resource of experimentally supported RNAs with both protein coding and non-coding function. So when we look at, so the first thing we do is compare like our complete data set with their complete data set. And it turns out that there are only 442 RNAs which are common across both the data sets. And then out of these 442, our models are able to identify like 229, but not identify 213. So still a pretty good overlap. And then we also compare to ribosome profiling datasets. So the thing with ribosome profiling is that there can be false positives, and then people have developed some methods to be able to find true positives from ribosome profiling data. And these methods include floss, ORF score, and phylo-P. And when we look at how our models compare to these, we see that we have high overlap with floss, highish overlap with ORF score, and not a very high overlap with phylo-P. So 2 out of 3, still pretty good. And then what we did was we decided to aggregate other evidence for every coding lncRNA that we discovered. So so we ran BLAST and tried to look for mass spectrometry data, also ribo-seq, and other coding potential prediction tools, and then finally AlphaFold. So here is the data. So for example, for this RNA, it's a long non-coding RNA and with a specific biotype of long intergenic non-coding RNA. And according to some ribo-seq data, like we are pretty confident that this is bound to ribosomes and likely being translated. And then Peptide Checker is a tool that analyzes mass spec data publicly available, and that also has a high confidence saying that I think this is being translated. And then when we look at BLAST, we find a very significant hit with an existing protein. And then one other thing we do is compare with other coding potential prediction tools. So coding potential prediction tools are like you give them an RNA and they give you a probability, like what is the probability for this being a coding or non-coding RNA. And as you can see, for CPC2 and CPAT, there is a high, like they predict that this RNA has a high coding probability and the third one does not. And then these are the predictions of our models. So when we look at the mean probability across different epochs, our models like Transformer and LSTM seem to think that this might be a coding RNA, but CNN doesn't. And finally, when we extract the ORFs present in this RNA and then ran AlphaFold on them, we see that according to AlphaFold, we are like, it folds into an alpha helix, and the confidence of this alpha helix fold is pretty high. So like aggregating all of these different kinds of evidence gives us a better idea. Like it helps us say more confidently that this lncRNA might actually be coding. And here I put another example, and the difference here is that even though we get from ribo-seq and from mass spectrometry and according to BLAST hits, we are pretty confident that this RNA is likely translated. Other coding potential prediction tools, they don't think that this is being translated. So this shows that our models can predict coding probability better for some of these RNAs as compared to the existing tools. And again, with AlphaFold, when we look at the ORFs and we see that there is a very high probability that this falls into an alpha helix. And the final thing I want to talk to you about is clustering of the embeddings. So what happens is that people have developed some methods to analyze nucleotide sequences so that if you look at the length of the ORF and translate the ORF and see how stable the protein is. So people have developed features like that. And using these features, when we cluster the RNAs, it seems that the coding RNAs are shown in blue again, and it seems that there is this region of continuity between what is coding and what is non-coding, and most of the possible misannotated RNAs that we identify fall close to this region of continuity. So I guess the idea here is that the categories of coding and non-coding might not be as different as we used to think that they are, and there might be some continuity between the two. So it might not be just two classes, but more of a continuum. So, in conclusion, it's possible to identify misannotated lncRNAs based on nucleotide sequence by leveraging the training dynamics of deep learning models, as I have shown you. And this work offers promising potential for assisting experimental methods in characterizing the hidden proteome encoded by misannotated lncRNAs, and also for curating better datasets for building coding potential prediction tools. And that's the end of this talk.
Much for this nice presentation. We already have some questions at the chat box.
Let me look at those. It will change, but can everyone see the questions, or is it just...
Yes, everyone can see the questions.
Okay.
Chat.
It will change based on the threshold or epoch numbers. How do you decide the threshold or right epoch at this point? Using some very well-known ones. It reminds me of the Chandra decision. Yeah, the way we decide how many epochs to run is by using standard practices from the machine learning field. There is something called early stopping. So you take into account like how much your model is learning, and at some point it starts plateauing the loss or the accuracy if you're following that. And before it plateaus and starts to overfit, you stop the training, and then that's how many epochs you train. So for the first one, we use stuff like that, early stopping, to make sure that to decide on how many epochs to run. All right. Some lncRNAs might have some coding regions. Do you think this might be contributing to the issue of identification and separation of these? I guess this was asked in context to some particular slide. I'm not sure I get that question. So if you want to ask that, I would be happy to. If you want to, yeah. I don't understand the question. All right, I'll move to the next one then.
If right now, if you are writing a question, please use that little hand icon down at the chat box so that we will know somebody is writing a question.
Okay, I'll move to the next one. I'm wondering if there is any future or ongoing plan for separation among long non-coding RNA subspecies during the decision process. All the data we have is only for humans, so I guess that question, like, we don't need to do that because we have just human data. First question box is answered. Okay, I'm a little curious. How do you make use of using 3 different algorithms you have described? Is it to increase confidence during the decision process? Yeah, that's exactly it. So I showed you one of these slides where we look at the overlap between the 3 different models, and even though a lot of them overlap, there are also some that do not. So it's just to give us a better idea of, like, it's better than using just one model. Okay, I mean the separation of long coding versus coding. Okay, I guess that's it, unless there are any more questions.
Okay. I guess we don't have any other question. Thank you so much, Afshan, for accepting our invitation and for giving us for this speech.
Thank you for the invitation, and it was really nice to be here.
Yeah, we would like to see or hear from more from you in the coming years, hopefully.
I hope so.
Yes, you can get the recordings. We are gonna share it on YouTube probably by the end of this month. And I think we can right now end the webinar. Thank you so much for joining, everyone. And thank you so much again, Afshan, for giving this nice speech. I wish you all a good night, a good evening, and thank you for joining.